Hello everyone, and welcome back to my channel where we learn everything science. My name is Aubrey Molln. I'll be your presenter today, and today we have a very interesting topic. We're going to be talking about the microbial infectious disease called tuberculosis, also known or abbreviated as TB. So, put on your thinking caps and let's get to it. So before we really get into the meat and potatoes of this presentation, I just kind of want to go over what our main objectives are as we go through this topic. So as you can see over here, I kind of have a table of contents of the order in which we're going to discuss different parts of tuberculosis. So for instance, we're going to have the taxonomy, general information, worldwide impact, and you can just kind of see going down the line what we're really going to be talking about. The big one is going to be the physiology and the pathogenesis of this particular disease. And then as we progress, we're also going to look at major signs and symptoms to look for, how people can become screened for TB, and how a diagnosis and treatment protocol will occur if one is found to be positive for tuberculosis. And then at the very end, before we cap off this presentation, I'm going to kind of talk about a very relevant research experiment that relates to this infectious disease and some breakthroughs that have occurred in the last few years. Okay, so as I mentioned at the very beginning, the disease in which that we're going to be talking about in this presentation is tuberculosis, also abbreviated as TB for short. And some people might be wondering, what is that microbe or the bacteria that actually causes this infectious disease? And the answer to that is actually Mycobacterium tuberculosis. It's right in the name. So it was actually discovered by Robert Koch. He announced the discovery that Mycobacterium tuberculosis caused tuberculosis on March 24th, 1882. And in that year or around that time period, tuberculosis was running rampant. About 1 in 7 people would contract the disease, and many of those victims that contracted it would die. So it was a serious disease. I mean, it still is serious today, but at least today we know more about this particular infectious disease, and we also know how to treat and or prevent it. And it's an interesting thing because on March 24th is the day that he officially announced his discovery. And today, that same date is now known as World TB Day. Now, that's not a celebration of it until it's completely eradicated. But this day is actually dedicated to help educate the public about the worldwide impact of TB and how to better help prevent the spread of it. And a few little quick facts here that I wanted to include. It's important to know that TB is one of the top 10 causes for childhood mortality. Now, that is saying a lot if it's in the top 10. And as you can see here, the statistic is just mind-boggling, at least to me. As we kind of go down, we kind of see the impact that this particular disease has. It affects 1/3 of the global population, and 1.3 million TB deaths are accounted for in Australia and South Africa, just those two countries alone. So that's pretty significant. And as I said, 1882 is the year that the discovery was made and announced. Another quick fact is that 12 months of treatment is to be given for serious forms of TB, because you can have mild versions and you can have serious versions. And then also, a lot of times people who have tuberculosis don't realize they have it until it becomes more significant or pronounced. Most of the time, people who smoke, for instance, 25% of them just think that the cough that's non-productive is caused from years of smoking, when in fact they actually have and could be spreading this infectious disease. So in the last slide, we talked about how Mycobacterium tuberculosis is the species of bacteria that actually causes tuberculosis. And I just wanted to kind of have the breakdown or the taxonomy, its classification in the biological world. Many of you should already be familiar with the kingdom, phylum, order, suborder, family, genus, species of taxonomy hierarchy. Some people might know the mnemonic, which is King Philip came over for good soup. You may have heard different versions of that, but I always think that it's a good idea to have the breakdown of any sort of biological organism that we're talking about. It kind of shows you the breakdown on how it's classified, going from very, very broad in the kingdom section all the way down to extremely specific for the species itself. So in this presentation, Mycobacterium tuberculosis is the species of bacteria that we're truly going to be focusing on. Before we talk about the physiology of Mycobacterium tuberculosis, I kind of just wanted to throw this slide in to show you the worldwide impact of TB, where it affects, where it's concentrated at, and just a few more statistics. Over here is the world. I had mentioned before in our fast facts that about 1/3 of the population will contract tuberculosis, not necessarily die, but they will contract it and or spread the disease. As you can see over here, we have a world map of the number of cases by density. You can see here that the darker the color is, the more cases were seen or tested for. And as we had said, about 1.3 million cases were recorded in just South Africa, which you can see is one of the darkest areas, as well as in India. And there's other parts too that are more impacted by the effects of tuberculosis, usually more in your developing countries. But I thought this would be a very interesting slide to include in this presentation, just to help you see the worldwide impact that this infectious disease can have. Also, I have some more statistics over here. About 9 million people will develop active TB. And that 1.3, or you can round it to 2 million people, will actually die from TB annually. That was back in 2012, and it has both curbed and increased at this, at different times. It's kind of goes in a wave. For a long time, when we had the discovery, the trend was going down from that 1 in 7 people of contracting it. It was going down, but then it reemerged sometime in the late 1990s, and by 2012 there were still 2 million people dying from this annually. So very infectious disease. It kind of makes sense looking at this both visually and just kind of taking in its impact. Based on our last slide, we can tell the infectious behavior of this particular disease and that it has the capability of spreading. But how does it spread? What is the mechanism that it uses in order to infect new people or more people? And the biggest way that tuberculosis is spread from person to person is actually through respiratory droplets. So that means that if somebody is infected with this Mycobacterium tuberculosis bacteria, and they develop active tuberculosis, if they are to stand really close to somebody else who is not infected yet, but then the infected person coughs or sneezes or transfers some sort of respiratory fluid to the other person, and it gets into their respiratory tract, so if they inhale it, then they could contract tuberculosis. So since this is an airborne disease, it can be very, very infectious, and so it can spread rather quickly. So in the past few slides, some of you may have picked up that I've been saying active tuberculosis, and you might be wondering, well, what's the difference? What is active tuberculosis? And before we move on, I want to make sure that we're all on the same page here, that there are two main types, or basically one type, but it has two phases that it could be in. One is a latent infection and one is an active infection. We saw from the world map and from the statistics that this particular disease is very infectious. However, The vast majority, about 90 to 95% of those who become infected with tuberculosis, their immune systems can actually contain the infection and keep it from multiplying. In other words, the TB disease remains latent or dormant as opposed to active, which usually causes symptoms and can easily be transmitted to others. When the host immune system becomes compromised due to something like HIV, malnutrition, old age, etc., TB can actually become reactivated and become very serious, especially if the infection spreads through the body. Moreover, people with active tuberculosis can easily infect 10 to 15 other people just being in close proximity to them within a year. So the biggest difference is the latent infection doesn't really spread, it just kind of infects the one host, while active infection can be spread to others. All right, now we're getting to the physiology of the Mycobacterium tuberculosis bacteria itself. So this mycobacteria is actually very slender and rod-shaped, as you can see in this reference here. And it needs high levels of oxygen to survive, which actually is what we call a strict aerobe. They possess a very waxy cell wall that is capable of retaining dyes even when exposed to alcohol. So this makes staining very, very easy to see. So as a reference, we call this the acid fast, which appears bright red in color, and it really highlights those rods when a Ziehl-Neelsen stain is used. And the wall also makes them incredibly hardy. So this allows them to resist weak disinfectants and survive on dry surfaces for months on end. So they're very, very resilient. Okay, so now we know what the bacterium or Mycobacterium tuberculosis looks like, and we know that it can enter the body through inhalation, and that's how it spreads from person to person. But what happens once it makes it into your body? So into your throat, you've inhaled it, where does that bacteria go and what happens? And so just as an example here, it's interesting because when we're looking and breaking this down, we realize that the body actually breathes in a lot of viruses and bacteria over time. However, our bodies have created defense mechanisms in order to help keep us healthy. One way that this happens is when the bacteria is breathed in, the air up here in the upper airways is actually very turbulent. What happens is this turbulent air will push the bacteria that's made it into your body up against the sidewalls of your trachea, which actually are coated in mucus, which is this green kind of slime here in this representation. When the bacteria becomes stuck into this mucus, there's little cilia that line the trachea that actually push the mucus up and out of your throat. So when you feel really sick and you realize that you're hacking up phlegm or you're coughing, that is your body's way of trying to get rid of the mucus with the bacteria in it, or just basically trying to get rid of the bacteria so it can't make you more sick. Now, if we go over here, the thing about tuberculosis bacterium is that it's able to avoid the mucus trap, which a lot of other bacteria get caught in. And this isn't good for you because what happens is that means it made it through the upper airways and makes its way down deep into the lungs. And it usually settles in the alveoli, which are those grape-like structures that we've seen all through biology classes growing up. And the thing that we're going to be focusing in those grape-like structures are what are called macrophages. So macrophages are actually the white blood cell or the defense cells that get rid of bacteria that have made it into your body. It's another line of defense, you could say. And macrophages are what are called phagocytosis cells. And that's because they perform what is known as phagocytosis. What phagocytosis is that when the bacteria comes in contact with the macrophage, it'll actually engulf and try to digest the bacterium to get rid of it. And it does this with the help of a lysosome. In this particular illustration, you can see this right here is the lysosome. The bacteria has been encapsulated by the macrophage, and one of two things can happen. Usually when the bacteria is able to digest it, the lysosome will release what are called hydrolytic enzymes. That's basically enzymes that can break down the bacteria, and the macrophage can digest it and move on to the next bacteria that's in your body. Now, the other side of the coin is if this is tuberculosis bacterium, what happens is something quite different. Instead of allowing the lysosome to release these hydrolytic enzymes, the bacteria will actually release a protein that blocks these enzymes from being able to be released. That means that the bacteria doesn't get broken down, and in fact, it what we call proliferates. So that means it multiplies and creates what's known as a localized infection. At this point, somebody has started to develop what's known as primary tuberculosis. This means that the person will start to show signs of the infection soon after being exposed to the TB bacteria. Now, even though it sounds really bad, most people at this stage are actually asymptomatic. That means that they don't show any signs whatsoever. Others, on the other hand, will show more flu-like symptoms that are pretty mild. However, about 3 weeks after the initial infection, cell-mediated immunity kicks in, and this is where immune cells actually surround the TB infection and creates what's known as a granuloma, which essentially is an attempt to wall off the bacteria and keep it from spreading. The tissue inside the middle then dies as a result, and this process is called necrosis. This means cheese-like necrosis, since the dead soft tissue is kind of yellowish white in color, very soft, and kind of resembles that of cheese, as you can see here as a visual comparison. And when necrosis happens in the center, this walled off granuloma then becomes a gone focus. So as these gone focuses are happening and the body is trying to retain and keep the spread of the bacteria in check, what happens is that the tuberculosis can actually spread to what's called the hilar lymph nodes. They're either carried over there by the immune cells through the lymph, or a direct extension of the gone focus infection causes caseation. And together, these casating tissues and associated lymph nodes actually make up what's known as the characteristic gone complex. So gone complexes, this is what it looks like in the lymph node. The gone complex is what it looks like over here. They're usually subpleural, meaning that they're located in the lower part of the lungs. And the interesting part is that they are encapsulated by the granuloma that undergoes fibrosis and often calcification. This means that it produces scar-like tissue that can be seen on x-rays. And I actually included an example here. You can kind of see those kind of white splotches. That's what scar tissue looks like in an x-ray. And this is basically a breakdown of what the calcification looks like in a cartoon manner. And this is what it looks like in real life. The interesting thing is that the scar is a leftover remnants, and the mycobacteria is killed off by the immune system, and usually that is the end of the infection. However, that's not always the case. In other cases, what could happen is when the bacteria becomes walled off, it can remain viable, and that means it's still alive. It's just in a dormant state. That means it can't spread the infection now, but it has the ability to spread the infection in the future if triggered. Now, this trigger can be something as in the immune system becoming compromised in some way. This means that if the person contracts HIV or is malnourished or has been advancing in age, all of those are conditions that can actually compromise the immune system. If this is the case, then the gone focuses actually become reactivated and the infection spreads to one or both of the upper lobes of the lungs. Now, why the upper lobes? It's thought that the upper lobes actually experience the most oxygenation. Now remember, the Mycobacterium tuberculosis bacteria are strict aerobes. That means that they need oxygen to survive. So they go to the place where oxygen is most available, which in this case would be those upper lobes. Now, at this time, when it's kind of stewing around in those upper lobes, what happens is that if the person has been exposed to this bacteria before, which if it was in its dormant state, that means it was, memory T cells are actually what recognize previously exposed bacteria. They're called memory cells because they retain a memory or an imprint of the bacteria that it has fought in the past. If and when I should say, it recognizes these bacteria, it releases what are called cytokines. These cytokines try to control the new outbreak. And this can actually form many new places of what's called casus necrosis. That's those yellow splotches, as we've seen, it kind of looked like cheese in the previous slides. Except this time, what happens is that these casus necrosis actually cavitate. This means that they form cavities where the bacteria can disseminate or spread through the airways and lymphatic channels to other parts of the lungs. So if it were to spread to the other parts of the lungs, it can actually cause what's called bronchopneumonia. Another thing that it could spread to is the vascular system. And if this happens, it can actually infect other tissues of the body. If this happens, this is what's called systemic miliary tuberculosis. So what is systemic miliary tuberculosis? This is when the TB has spread to other tissues and can cause complications related to other organisms that are affected. So in this slide, I just kind of listed a bunch of organs that could become affected and what associated disease could propagate from that particular exposure. So a few would be if it were to make it to the brain and infect the meninges, it can cause meningitis. If it were to infect the joints, it could create what's called mycobacterial arthritis. If it went to the kidneys, it could cause steropiria, which is an increase of the white blood cells in the urine. And these are just many different examples of how infectious and how debilitating tuberculosis can be on the body. It can create all these problems if it makes it to the vascular system. So it's always best to try to catch it early because if it reaches this stage, it is so much harder to treat. So the next question would be, how can somebody tell if they have active tuberculosis or not, whether it's mild or severe? So obviously, depending on the severity, it's going to have these signs and symptoms that one experiences vary. But some of these are pretty regular for all types. So for instance, a lot of people with tuberculosis have flu-like symptoms that usually entails fever, night sweats, fatigue, and chills. As the tuberculosis progresses, some more severe side effects would be unintentional weight loss, loss of appetite, and then you could also have breathing difficulties or pain. And then when it gets to a higher severity, what typically patients experience for symptoms would be coughing, sometimes non-productive, sometimes productive. That means that you've actually coughed up mucus. They experience that for 3 or more weeks, so it's a cough that just won't go away. And then another one is coughing up blood. If you are experiencing any of these symptoms, it is highly recommended that you go see a doctor. Typically, if a patient were to show up to the doctor's office experiencing or showing signs and symptoms that were shown in the previous slide, then the doctor will most likely perform a test called a tuberculin test, also known as a Manteau or a TB test. This particular test is actually a purified protein derivative, or PPD, intradermal skin test. That means that tuberculin is injected between the layers of dermis on your forearm. Tuberculin is actually a compound of the bacteria. And if a person has been previously exposed to tuberculosis, the immune system will react to the tuberculin and produce a small localized reaction between 48 and 72 hours. If the reaction causes a large enough area of what's called induration or redness, as you can see in this live picture over here, it's what's considered a positive test. Now, a positive test, one needs to keep in mind, just means that the patient has been exposed to tuberculosis in the past. That doesn't mean that what they're currently experiencing is active or latent. As an alternative to tuberculin skin test, there's also what's called an interferon gamma release assay, or it's called IGRAs, or IGRAs for short. This is where they look for evidence in the blood of previous exposure to TB proteins. Since this one's a blood test, you don't need to show up again to have the test read to you like you do for the PPD test. The IGRA is more specific to tuberculosis rather than other types of microbacterial infections, and is unlikely to be a positive as a result of having what is known as BCG vaccine in the past, a vaccine that actually can help protect against TB. And this is a very useful feature of an IGRA, since BCG vaccines is given to many children around the world to help prevent the spread of tuberculosis. Also, it's important to remember that if a person has screened, whether it's using the PPD test or an IGRA test, anyone with a positive result will typically be sent for a chest x-ray to look for signs of active tuberculosis disease. Once a chest x-ray is performed, it's very important for medical staff to collect samples from either the sputum or via a bronchoalveolar lavage, which is where a bronchoscope is actually inserted through the mouth or the nose into the lungs, and fluid is squirted into that area, as you can see over here, and then the fluid is then collected, ta-da, in your little beaker. These samples can then be sent to a lab for staining, cultures, PCR, and other tests to look for evidence of that Mycobacterium tuberculosis bacteria. Depending on the type of tuberculosis you have will really help determine what type of treatment options are available to you. For instance, if you find that you have a latent infection of tuberculosis, that can typically mean that the treatment option will be the taking of a single drug for a prolonged period of time. The most common one would be a 300 milligram dose of isoniazid every day for 6 to 9 months if you're an adult or an adolescent, or 5 milligrams a day if you're a child. However, if somebody finds that they have an active tuberculosis disease, it typically requires a combination of antibiotics. And this is what is typically called the RIPE regimen, R-I-P-E. And usually it takes patients a few weeks to become non-infectious to others. Until that point, this patient can actually spread the TB to others, and it's typically adults with reactivated TB that is the most infectious. This being the case, patients are typically kept in a negative pressure room, and visitors are asked to wear a protective N95 mask that can filter out oil aerosols. N for not resistant to oil, but can filter out 95% of other aerosols, in this case, tuberculosis. Even after patients are no longer contagious, they're typically kept on multiple medications for many more months to make sure that the bacteria is completely destroyed, usually with directly observed therapy, or DOT, where somebody actually watches and confirms that you are taking your medication. On top of all of this, there is additional worry about new drug-resistant strains of tuberculosis that are changing infections in various parts of the world. You may have heard of MDR-TB, which stands for multiple drug-resistant tuberculosis, and the other one is XDR-TB, which is extremely drug-resistant tuberculosis. Both of these variants are very hard to treat, and they don't die in the presence of your usual antibiotic regimens. The bottom line is that to get an effective treatment, it's very important to make sure that the drugs being used will work against the specific strain of tuberculosis, that the multiple medications are used together to prevent the drug resistance from developing, and the medications are used for the entire course of the therapy so that all of the Mycobacterium tuberculosis is killed off completely. So what are some preventative measures that a person can take to avoid contracting tuberculosis in the first place? Well, the number one thing is to get vaccinated. Young children are being vaccinated around the world with BCG vaccine. This is to help them from contracting TB from a young age. Some other good practices to take part in is practice good hygiene, have healthy exercise and eating habits, ventilate your house, stop smoking if you're a smoker, and avoid prolonged contact, especially with those experiencing symptoms of being sick, whether it seems like tuberculosis or even the common flu. All right, guys, so we've finished more so the main concepts about tuberculosis. And so I just want to take this opportunity before I end this presentation to kind of go over a very interesting article that I found that is relevant to tuberculosis. And the article title is actually What Animal Models Teach Humans About Tuberculosis. It's an article published in 2008 in the Journal of Respiratory Cell and Molecular Biology. And I think it's a very interesting topic because the authors are basically looking for, of the three main lab animals used in animal trials regarding tuberculosis, which are mice, rabbit, and guinea pigs, what are their pros and cons, and ultimately which one would be the most beneficial to use in future tuberculosis studies. And it's very interesting to see what they were able to compare. For instance, they were looking at anything from maintenance of the animal, so taking care of it. Are they high maintenance? Are they low maintenance? What is their time period for reproductivity so they can do more trials? What is the size? Because it's really hard to house a large animal in a small lab environment. It's things like this that they review throughout their journal article. For instance, when they're looking at mice, they see that mice reproduce very quickly, so they can do a lot of trials. They're small, they're easy to manage, and they're relatively resistant to the TB infection. Along that same line, rabbits and guinea pigs are more vulnerable to the infection than mice are. What's also fascinating is that this article also talks about how Robert Koch, the discoverer of the Mycobacterium tuberculosis bacteria that actually causes tuberculosis, in his initial studies, he used guinea pigs because of their vulnerability and susceptibility to contracting TB. So over the years, mice and rabbits have been added into these lab practices. In the end, the authors found that mice were most often and most suitable for immune responses to TB infection due to their resistance, as well as drug regimens against TB. They also found that rabbits are very good candidates for developing cavitary TB and offer a means of study to the factors leading to this form of disease. And then you have guinea pigs, since they are the highest vulnerability of the three. They are very ideal for studies of airborne transmission and vaccine efficacy. So really, all three of them were found to have benefits. And it seems that It really depends on the type of study that one is performing before they can choose which animal host to use in their experiments. Like I said, I found this article very interesting. They went way more in depth on the details, but if you are interested, please feel free to look it up. Again, the article was published in 2008, and the title is What Animal Models Teach Humans About Tuberculosis. Very good article, highly recommend. And there you have it. Thank you so much, you guys, for joining me with this presentation. I know it was long. I kind of nerded out. I really went in depth on this one, but I hope that you enjoyed it. I have here my first page of references that I use. The highlighted reference that you can see right here, that is the article that I kind of talked about regarding the animal models. If you need the information to look it up, it is right here, so feel free to pause your screen so you can type it in and look at it at your own convenience. Since this presentation was so long and it covered a lot of material regarding tuberculosis, I used a lot of references, as you can tell, so this is page 2. And there you have it. Again, thank you guys so much for hanging in there with me. If you made it to this part, I hope that you learned something and can take away a lot of knowledge about tuberculosis, anything from what causes it to how it actually manifests, to different signs and symptoms to look out for, all the way to different preventative strategies that you guys can use to protect yourself and your loved ones. Again, I hope that you guys can take something away from this presentation, and I hope to see you again next time. Until then, have a beautiful day.